Hello everyone, myself Ovik, PhD student in SN Bose National Center for Basic Science under an autonomous institute under Department of Science and Technology, Government of India. Title of my talk is Microscopic Insights to Relaxation Phenomena in Protein. At first, what is relaxation phenomena? Relaxation phenomena is time-dependent change of any physical quantities following external perturbation. Now, why it is important? Now, relaxation phenomenon is a good probe for information on the local environment of the system. Now, microscopic understanding of the relaxation phenomena on biological system is not as typical as any other condensed matter system. Just because the biological system involves extended range of time scale and large number of degrees of freedom. Now, if you consider biomolecules like protein, this is a cartoon picture showing the range of time scale associated with the protein motion. So here you can find femtosecond bond vibration to the domain movement in a microsecond time scale. Now, just to set the stage, what is a protein? We all know that protein is a polymer chain where each monomer is basically amino acid sequence. This is a cartoon picture of protein primary structure, that's the sequence of the amino acids. This is protein secondary structure, which basically consists helix, sheet and loop, and these are proteins tertiary and quaternary structure. I am skipping this. Now, if we consider the degrees of freedom, then in protein case the most important degrees of freedom is dihedral angle. So what is dihedral angle? Dihedral angle is angle between plane. So if you zoom in the protein backbone, so this is it. If consider C and C alpha atom, they constitute plane, and N C alpha C, they constitute another plane. The angle between them is phi. Similarly, N C alpha C and C alpha C N, angle between them is psi. So these two are one of the most essential degrees of freedom in case of protein. So why we are considering only this? Because protein has phi 1, chi 1, chi 2 and all those. So I will discuss one by one. Okay, now protein relaxation may happen due to presence of ligand, sometimes due to altering solvent conditions, sometimes due to change of thermodynamics properties. Now, protein relaxation typically includes the conformational fluctuation. Now, one can probe the conformational fluctuation experimentally using method like NMR and X-ray. But the problem is NMR and X-ray basically give an ensemble picture, but protein system is inherently dynamic. So if we need to understand the dynamics of the system, then we need to depend on the computer simulation. So basically all atom molecular dynamics simulation. So in my work, we probe the static and dynamic aspects of conformational fluctuation of protein to understand the relaxation phenomena microscopically in terms of protein dihedral angle. So just in two lines, why we are using the dihedral angle. So coordination between various functional parts is essential for any kind of biophysical or biochemical phenomena inside the cell. Now, earlier theoretical report based on that, they told that coordination can be best understood in terms of dihedral angle. Even one can estimate free energy and entropy from the population of different value of dihedral angle. So dihedral angle is important. Now, generally we use the molecular dynamics simulation technique, which I will not discuss here. So this is the general method of the molecular dynamics, and this is the form of the force field. Okay, so in my first project, what we try, we all know that dihedral fluctuation is not directly amenable to the experimental probe due to the time scale, because dihedral fluctuation occurs in nanosecond time scale. On the other hand, NMR can probe dipolar fluctuation in terms of cross-correlated relaxation rate. Now, what is CCR rate? CCR is basically given by spectral function of dipolar fluctuation. Now, the issue is time scale. Again, the dihedral fluctuation occurs in nanosecond time scale, whereas CCR gives integrated information over time on dipole orientation correlation function. So these are general form of the spectral density, and this is the time dependent correlation function. Now, what do we address? Now, dipole fluctuation at the level of nanoseconds obtained from simulation, how much contribution it made to the experimental observation. First we probe this thing, and then we discuss for dihedral fluctuation how corresponding spectral function obtained from simulation is comparable with the dipolar fluctuation obtained from simulation. Because again I am telling that issue is time scale, so relating both two things is not a trivial. So that is our approach. So we consider protein GP3 and protein ubiquitin for our system. At first we calculate the time dependent correlation function, considering the dipole generally people use during the NMR experiment. So we consider H and N and C alpha H alpha dipole inside a protein. That's an intra-residual case. Even people use sequential also, so we also consider that case also. So this is a time dependent correlation function for I7 residue of Gb3, and this is for sequential dipole, that two dipole H and N and C alpha H alpha in two different residue for Gb3, and instead show the short time decay. So from that we can calculate the correlation time scale. So if you check the histogram from Gb3 and from ubiquitin, you can find that correlation time scale of dipolar orientation is approximately nanosecond, and this is a correlation plot for, this is for inter-residue and this is for inter-residue, that is sequential, that is two dipole in two different residue. And when we compare this plot with the experimental CCR, we find a good correlation. So this is for inter-residue, this is for sequential. So from that what we can conclude, the short time decay of dipolar fluctuation, what we will describe the experimental CCR. Even for distant residue, initial decay of dipolar fluctuation can capture the experimental CCR. Now we similarly plot the dihedral time dependent correlation function, which shows similar kind of trend having correlation time scale in nanosecond. Now this plot shows the histogram of the correlation time scale for dihedral. We can find the dihedral fluctuation occur in nanosecond time scale. Next, we relate a spectral density of dihedral obtained from simulation with the spectral functioning of dipole obtained from simulation. And here we do the analysis similar, we plot the similar correlation plot, but here we use that plot is done based on secondary structure, because we find some sensitivity based on secondary structure of protein. So we consider Gb3 and ubiquitin together because they are same kind of perfectly ordered protein. So this AB is for helix, CD is for sheet, and EF for loop, and this is for correlation coefficient. And what we found, that correlation coefficient moderately well. Even we probe the internal dynamics of the protein, and this can be probed using if you align the structure with the initial one, then we can prove the internal dynamics also, and there also we are getting the correlation. So overall the dihedral and dipolar fluctuation show moderate correlation for this protein, and we find good correlation for dipole fluctuation in the sequential residue also. So I did not show the sequential result here, but we are getting good correlation for that case also. So what is the conclusion? So nanosecond time scale dipolar fluctuations dominate the CCR, and secondly that's CCR that which we calculate from the experiment, and dipolar fluctuation is correlated with the dihedral fluctuation in terms of spectral density. The work is published on Chemical Physics Letter in 2022. Okay, so in next project we probe the conformational fluctuation in the molten globule state of alpha protein, alpha lactalbumin protein, where we check the conformational fluctuation of the protein due to altering the solvent condition. Now what is molten globule state? So molten globule state is an intermediate state between protein native state and unfolded state. Now why it is important? So many of the protein in molten globule states are known to be functional. Like if you consider bovine lactalbumin protein, in molten globule state this protein calcium ion come out from the protein and it bind with the oleic acid and it form Bamlet like complex. Now the problem is protein and this Bamlet like complex actually shows cytotoxic activity against the cancer cell line. Now the problem is protein and molten globule state are highly dynamic and flexible, and that's why due to lack of crystallization, molten globule state conformation and their functional significance are not well understood till now. On the other hand, if you consider intrinsic disordered protein, we found that generally what people found that the disordered protein inherently possesses the metastable conformation. So metastable conformation we all know generally in terms in physics and chemistry. Our point is the disordered protein has not any lack of structure, they have not any proper structure, but they are functional. Even they have not structure, they are very much fluctuating. Similar case is happening also in protein molten globule state. So what we address whether the conformation fluctuation in the molten globule state has any similarity with the intrinsic disordered protein. Okay, so what we take? We check the bovine lactalbumin protein both in hollow and apo state. Apo means there is a calcium ion. So at first we check whether we are in a molten globule state or not. So one can check that thing using some RMSF calculation, radius of gyration, native contact and non-native contact. People did work in this simulation earlier result, and we check this thing that this is a molten globule state is an intermediate state between protein native and unfolded state. So that's why a radius of gyration must increase. Native contact that should be decreased, and non-native contact should increase. So we check that thing, that we use the pH2 as a known. Okay, so we get a molten. One can get the molten stable state using at a low pH or high temperature, sometimes due to presence of some kind of solvent like urea, all those. So why we use the constant pH molecular dynamics to maintain the pH of the system using the amber basically. So this is a radius of gyration increase at pH2, native contact decrease. So this is on molten Gould state, and non-native contact increase. So we are in the molten Gould state. And even RMSF is very much fluctuating. Even if you check the principal component analysis of the C alpha atom, you can find that at pH2, that is known to be molten Gould state, their fluctuation and the contribution of species is very high as compared to neutral. So what is the conclusion overall? That the protein and molten Gould state highly dynamic and flexible. That is known from earlier result and experiment also. So next what we did the dihedral principal component analysis that was developed by the Gerard Stock, and we use from that dihedral principal component analysis, we using the PC125, we check the negative log of the histogram of that dihedral principal component, and we can find the metastable nature. But PC629 metastability decrease, but PC125 there is a large metastability. So this is for molten Gould state, that's a pH2. But if we consider neutral case, metastability is less as compared to previous one. So this suggests that protein at molten Gould state have some metastability. Now our aim is to identify the metastable nature of the protein in terms of essential coordinate. So what is essential coordinate? Essential coordinate is shown to represent reaction coordinate that explain the mechanism of the underlying process. So what we have identified the metastable state using some kind of clustering like geometrical clustering, density based clustering both, and we identify the metastable state formed by that MD coordinate, that is that dihedral angle. Next we use decision tree based algorithm to identify decision tree based algorithm, that is basically we use the XGBoost algorithm machine learning model to identify the essential coordinate. Now best advantage of decision tree algorithm is during the decision making you can identify importance of the feature. So here is a feature is here basically dihedral angle, the MD coordinate. So at first you identify the importance of each coordinate, okay, then you discard one most least important coordinate at first, and you reiterate the model with remaining coordinates, and you check the accuracy. So accuracy means how many states they are this model, how many this machine learning model, how many states they predict properly. Okay, so that's the way of the checking accuracy. So from accuracy plot, if we discuss any non-essential coordinate, then accuracy remains same. But if we discuss any essential coordinate, then accuracy even fall. So during the last 10 iteration, we found this coordinate from psi 80 to 577, where psi 80 we obtained in the last iteration. So psi 80 is most essential coordinate. So we take first 10 essential coordinate, where psi 80, 579, psi 18, etc. Now the point is, what is the importance of this model? So if you consider psi 80 and 579, which are the most essential coordinates, they belong to calcium binding loop. So it is known that calcium binding region plays a vital role in molten Gould formation. Now this psi 80, this is known from experiment. Even from machine learning we are getting the same information, that psi 80, they are identifying the psi 80 as the most essential coordinate. Now next we check what is the conformation preference. So what we found, this is a conformation preference plot. So how much time they are helix or secondary or loop, helix or loop or seed. They became unstructured over the time, most of the time, but initially they belongs to helix structure. So that told that initially they have the structure, but during the simulation they lost the structure. So you may tell that this is a story of 10 essential coordinate. What about the others? Because I have 123 residue, that means 246 coordinate total, 246 phi. So I perform the structural persistence calculation, which done by Nilanjana Sengupta, and we use the structural persistence calculation based on dihedral angle, and we make a histogram of the structural persistence. So lower structural persistence means they are less structured. So what we found, the essential coordinate has the lower structural persistence, but non-essential coordinate has the higher structural persistence. So what is the conclusion? We conclude that we find a metastable state separated by energy burial in the molten Gould state. In essential coordinate initially belong to stable secondary structure, although they prefer unstructured conformation in the molten Gould state, And third point is molten cool state of protein can be viewed as induced disordered protein, not an intrinsic disordered protein. So I already told about the functionality of the protein of the at the molten cool state. They bind with the oleic acid and from the complex the bamlect, which shows cytotoxic activity against cancer cell line. But due to lack of crystal structure, microscopic understanding of binding is not yet explored. So we, in third project we did that work. So at first we identify the binding residue of the protein, which we actually bind with the oleic acid. So it is known that cleft region of the protein actually bind with the oleic acid, but not particularly which residues are actually responsible, it is not known. So what we used the conformation thermodynamics method, that is a histogram method, to identify the free energy and entropy change. So free energy is standard definition, where H is basically normalized probability distribution of dihedral angle theta, and H state max is basically peak of the histogram that represent the equilibrium value of this trihedral. So from that one can calculate the delta G, and even gives entropy formula one can calculate the entropy change. Now based on that we identified the residues which have the delta G and delta S greater than 0. So they are actually prone to bind. So these are the residue in the cleft region which have delta G and delta S is greater than 0, and they are basically prone to bind. So what we did, at first we identified the structure, representative structure from the simulation and molten cool state, and we use the clustering technique to identify the suitable structure, and we use the docking. So this is a oleic acid thing, and this is a dock structure, and this is the initial dock structure, and during simulation this is the, we can find the leg, the tail, hydrophobic tail part of the oleic acid actually go to the group of that protein. And after simulation, during the simulation, when we check the delta G and delta S for that active residues that are basically ordered. They are all ordered. Initially they are disordered, but when the ligand come, they are basically ordered. Now we also check the minimum distance between protein ligand. What we found, the minimum distance is nearly 3 angstrom, and there is always a hydrogen bond is always present there. So that support a strong binding between protein and ligand. Now this is a basically video showing the protein ligand binding. Next we identify the free energy. We identify the binding energy using the umbrella sampling technique. So this is umbrella sampling PMF plot for the protein at molten cool state. We find the binding energy is minus 8.3 kilocalorie, whereas from experiment it is minus 9.45. So it is very much similar. And we also do this for neutral case. Neutral case it is very less, because it is known that neutral case it will not go for binding. So it is minus 2.5 kilocalorie, that is within the experiment and within the error value. Okay, so next what we did, we treat the water molecule present at the surface of the protein behave differently than the bulk water, it is known. And interfacial water basically played an important role in mediating the protein ligand interaction. So what we did, we checked the role of water in protein ligand binding. So basically we calculated residence time, rotational diffusion and translation diffusion till now, and we consider 4 systems, that is system 1, water around protein in complex, system 2, water around ligand in complex, system 3, water around protein in free, and water molecules that are common to both protein and ligand. So this was done during the Sanjay Bandyopadhyay in JCP 2012. They show the similar thing for protein carbohydrate thing, and we use the same technique here. And at the given instant, water molecules that are found to be present near the protein surface within 6 angstrom are included in this calculation. So we consider the hydration water, only that water molecule which are within 6 angstrom. So this is a basically survival probability calculation, residence time correlation plot, dipole orientation. This is rotational diffusion. What we found, there are some water molecule, basically they are showing very slow dynamics, and that is considered for system 4, that is water molecules that are common to both protein and ligand. So what we conclude from here, that there is some water molecule which strongly bind with the protein and ligand, and they are basically show the restricted movement, and they are basically expected to play a role in complex formation. So conclusion, we address the microscopic behavior of the protein ligand and molten cool state. We explore the behavior of water and the interface, and third, we explore the heterogeneous influence of formation of protein ligand complex on the dynamics of those water molecules which are close to both protein and ligand. Okay, so in fourth project we developed the coarse grain model of the protein. So we all know that coarse grain description is basically lowering the representation from all atom to coarse grain, basically give us a well-known representation. So we build a bed spring polymer model to make a coarse grain description of the protein with a dihedral angle information. So these are typically, so here the amino acids we consider hydrophobic and hydrophilic amino acid. So each amino acid corresponding to one bit. So these are basically known bonded and non-bonded interaction between the bits. So there are two kind of bits are there, that is hydrophobic and hydrophilic. And the main part of the work is we institute the dihedral coupling. So at first we simulate the protein in all atom calculation, we simulate the all atom molecular dynamic simulation, and from that we calculate the dihedral angle, and from that we calculate the probability distribution of the dihedral angle, and now negative logarithmic value of that probability basically give the free energy value. So that is a intra-residual coupling. So similarly we apply inter-residual also, because we had two kind of bead, hydrophobic and hydrophilic. So there are four combinations possible. So in between hydrophobic, philic and philic phobic we are combined together. So there are three combinations. So for at any instant within the cutoff there are two type of bead may be possible. So red one is basically hydrophobic and green is hydrophilic, and total combination may be 4, that will be 4. So 3 into 4, 12 type of energy landscape may be possible, but there is possible. And this is a type of energy profile with hydrophilic inter-residue, and this is inter-residue. Now how this thing is included in the model? So we use the Monte Carlo simulations. We have the initial configuration XYZ coordinate and with phi psi value. So initial phi psi value is taken from the crystal structure dihedral angle. We give the random displacement and rotation degrees of freedom, calculate the change of energy of the system after that move delta E. Now delta E is came from this basically free energy profile. So for specific value of phi psi, what is the value of energy, and that's for inter-residual. Also energy is taken for the inter-residual responsibility from inter-residual, and also from this include energy is also included in delta E. Now if delta E less than 0, then there is a random number. So typically Metropolis algorithm we use. So based on that, this is energy plot, this is solvent distribution. What we found that, okay, so solvent distribution we absolutely find that hydrophilic peak is lower, hydrophilic peak is upper, hydrophobic is lower. And this is a R.C. Ramachandran plot, phi psi plot, where a circle represent the initial crystal structure dihedral angle, and triangle represent the average dihedral angle obtained from the coarse-grain simulation. That generally that is mostly similar. And we also did the dihedral principal component analysis to check the overall picture, and with the plot of PC1. So this is a PC representation of all atom, this is coarse grain. So what we conclude, we propose a simple coarse grain description of protein model where dihedral information is included, and these results are till now good agreement to the all atom description. So this work is not published, this is underway now. So conclusion is the microscopic relaxation of the protein could be amenable through the experimental probe, which might be useful to understand allosteric mechanism and impact on biological application. Binding affinity of the small molecular protein can have practical value in terms of potential application as a therapeutic drug, and coarse grain description is expected to explain complex phenomena inside cell which are not amenable to all atom simulation. So these are my last publication, where PCCP, this paper is that molten gold state and IDP that published in the PCCP, and these two are basically under preparation. So thank you for your attention.